Clinical Aspects of Viral Interference, a presentation by Professor Gershom Zaitchik, Faculty of Medicine, Hebrew University of Jerusalem. Baltimore classification places viruses in 7 classes or groups, and Baltimore classification has 2 interpretations. The says that virus controls cell and virus is a parasite, and the other that cell controls virus and virus is the message. Virus control cell should be abundant, it's false, and I use only the interpretation cell controls virus and virus is its message, and it is explained in several presentations. 7 processes triggered by 7 virus classes. Actually, Baltimore classification are 7 processes, and these processes, you have the virus which triggers the formation of provirus, and each class has a different provirus. So here's an example. We are going to talk about class 1 viruses. So here you have class 1 double-stranded DNA virus, which infects the cell and triggers the formation of the provirus, and it will also create the messenger RNA. So before we continue, let's define what's happening. Provirus is the set of cell genes with 3 functions. It stores the self of the cell, it controls who may infect the cell, and it controls virus production. And self is a set of provirus genes, unique to the cell. Virus is a cell messenger. Virus is created by provirus, and provirus stores self of the cell in its genome, and virus infects only cells with the same self. Again, self is a set of provirus genes, unique to the cell. So I discussed this idea in the theory of virus and cell, and you can check it there. So we are talking now about class 1, which represents 2 viruses, Blocks virus and vaccinia virus. So here is a patient who is infected by variola virus gets smallpox. A patient who is infected by vaccinia virus, he will get vaccinia pox, but he will never get smallpox. And this is known as Edward Jenner phenomenon. So we want to understand this Edward Jenner phenomenon in terms of the virology. So if we examine the class viruses separately, here you have class virus, which is variola, and we see that the virus attaches to a cell receptor, triggers the formation of provirus class 1, and as I said, the provirus controls the formation of virions, which are variola viruses. If we now investigate what happens to vaccinia infection, and so again, a vaccinia virus infects the cell, attaches to a receptor, triggers the formation of provirus class 1, and this provirus will create vaccinia viruses. So this is if you investigate the class 1 virus separately. But we are talking about infection by dependent viruses, which means that the vaccinia virus infects the cell, but it is a first infection because following this vaccinia virus, the cell will attempt to create variola virus, but so we have this vaccinia virus infect the cell as I described, and here the variola virus attempts to infect the cell in the same way, but it is blocked at the provirus, and the variola virus will never happen, never create variola viruses, so there were no variants. So this is the interpretation of what Edward Jenner discovered, and this is based on vaccinia interference with variola virus. So again, here you have the outcome of variola infection, you get smallpox. Here is the outcome of vaccinia infection, which blocks variola virus, and this will never get a smallpox. So conclusion, virus interference in the clinic, more to come, enjoy.